According to the Center for Disease Control, 1 in 68 children in the United States are born with an autism spectrum disorder. This is a 30% increase from 1 in 88 in 2012. As the number of children with autism grows, the need for finding better, faster, and earlier diagnosis is extremely critical. However, the average waiting time for a child to get a diagnosis is from 6 months to a year. Why? Lack of a firm biological marker for autism certainly is one of the reasons. What can we do to address this and make the diagnosis smoother and faster?
Hi, I'm Dr. Rajesh Kana. I'm a professor in the Department of Psychology. I direct the Cognition, Brain and Autism Research Lab. In our lab, we study the brain in people with autism spectrum disorder. We use different brain imaging techniques to understand the structure, function, and connectivity of the brain in autism. Our research lab has 2 main goals. Number 1, to identify a brain-based marker for autism. If we identify such a marker, we'll be able to diagnose autism earlier in life. Number 2, if there is a biological or brain-based marker, then how can we use training or intervention to target that to change the damaged brain? Our lab has several different projects addressing these goals. Ultimately, our aim is to help individuals and families affected by autism spectrum disorder.
My name is Martino, and I'm the project manager for the reading study in the lab. Many children struggle initially with reading. Children with sustained reading difficulties or diagnosed reading disorders, however, are generally at a disadvantage early in their education. For example, as many as 65% of children with ASD have struggles with reading comprehension. One of our projects in our lab addresses this topic. In the Brain Read study, or the Brain and Reading Enhancement in Autism Disorders, we identify children with autism who have difficulty with reading comprehension and provide a 10-week-long intervention lasting 200 hours for them and test the impact of this training on their brain using MRI scans before and after the intervention. Our preliminary data suggests significant changes in the brain's reading network in children with autism spectrum disorder as a result of the reading intervention. This intervention shows great promise in targeting brain plasticity in autism using cognitive strategies to affect change in reading comprehension.
Hey, my name is Julie Trapani. I'm a graduate student at UAB working as a research assistant and collaborator with Dr. Rajesh Kana on the Brain Read project. So this project and the reading intervention and brain-related changes post-intervention has far-reaching implications for kids with autism, and we're able to offer support and help for kids with autism across the country through this research study. So it's a pretty cool opportunity to be a part of.
I'm Megan Beckerson, a first-year Ph.D. student in the Cognition, Brain and Autism Lab. Adults with autism face a lot of difficulty in getting a driver's license and also while driving. In a project we call Transit, we examine how the social and executive brain in adults with ASD and ADHD respond while driving. This project involves simulated driving and MRI scanning. This will help us understand the brain circuitry involved in driving and will also help us design training to improve driving in disorders such as ASD and ADHD.
My name is Caitlin May, and I am a 4th-year Ph.D. student in the CBrA lab. Young adults with autism spectrum disorder struggle in social interaction. How do we train them to navigate the social world successfully? The Program for the Education and Enrichment of Relational Skills, or PEERS study in our lab investigates this. We are testing whether a 14 to 16 week-long PEERS intervention changes the brain structure and functioning in adolescents and young adults with autism. Findings from this study will provide valuable biological information about how a social intervention can change the brain and improve social behavior.